So this video is for chapter 9 on microbial control methods. The concept here is how do you control for microorganism contamination outside the body, right? Either on the skin surface or on other surfaces or fomites. This, I feel like, is one of the more prevalent chapters for those of you who are going into healthcare services, so nurses primarily, or PAs, right? People who are interacting with patients in a healthcare setting, and you need to consider how to reduce transmission of organisms, like from you to the patient, from the patient to other patients, or from the patient to you. So this chapter, I think, is actually one of the easier chapters to get through, in my opinion. So it's pretty straightforward. We'll go over some of these major concepts here. Ideally, in these lecture slides, in this lecture video, I can go over all of the concepts, but usually I kind of end a little early and have you guys just complete a worksheet on microbial control methods from your actual chapter workbook. So we'll see how far we get on these ones. The major topics that we'll cover with these slides are the categories of microbial control. So it's not just spraying down a surface with decontaminant or disinfectant. There are other ways that you can control for microbial contamination. What are those methods? And then the last section is on those chemical agents that you can use for surface decontamination or disinfection. There are some key concepts here. These are some particular vocabulary terms, so understanding the difference between sterilization versus disinfection, actually 4 of them. Decontamination and antisepsis would be the other 2, so we'll talk through that one in just a minute. And then what are those 3 main methods, and what are some examples for each of these things, using physical control, chemical control, or mechanical. So let's talk through this a little bit first, conceptually, right? When you have to disinfect or decontaminate, clean a surface, you need to consider what types of microorganisms you may be dealing with. So if you know that potentially there are spores, endospores in particular, that you're dealing with and you have to disinfect those, this would be if you are dealing with like a surgical instrument that's going to penetrate deep into the tissue. Spores really love deep tissue inside humans or other animals, and they germinate well there and can cause some serious infections. So under those circumstances, you definitely know that you need to eliminate endospores. But we also know that endospores are the most resistant life form of microorganisms. So because endospores are the most, and this is outside of something like prions, which we talked about in the beginning of the semester, those are not life forms, those are proteins. So outside of that, when we're talking about actual microorganisms and the more resistant form or genre of microorganisms, the endospores are the most resistant. And so the highest level of cleanliness that you can get is using a sporicide, something that kills endospores. And if you can eliminate endospores, essentially you can eliminate anything else, right? Because they are the more resistant. Underneath endospores, protozoan cysts, which are very similar to spores, right? They are very hardy also. Those are the transmissible form of these, these protozoa. Mycobacterium, if you guys recall from our laboratory exercise, mycobacterium have mycolic acid in their cell wall that make them really resistant to environmental degradation because it almost forms this waxy coating around the bacteria. Which makes them very resistant to microbial control, but also antimicrobial medications, which we'll talk about in the next chapter. Pseudomonas, one of the leading nosocomial agents. Remember, these are hospital-derived pathogenic infections. You'll find Pseudomonas everywhere in the hospital, and it's very challenging to eliminate Pseudomonas contamination. And also because they are so prevalent in the hospital setting, and because they're actually quite good at conjugation and transformation. So they really readily take up foreign DNA and adopt that genetic alteration that makes them highly, not mutated, but highly adaptable to altering circumstances. So they become a very resistant form of organisms. Naked viruses would be up here too. If you consider that, the difference between naked and enveloped, the envelope virus has an envelope, which is a cell membrane derived from the host, which is a lipid-based envelope, and therefore anything that disrupts that lipid membrane or lipid cell membrane would be effective at eliminating enveloped viruses from the environment. So naked viruses are more resistant than enveloped ones because of the propensity for lipid for dissolve. The least resistant down at the bottom of the list would be vegetative bacterial cells, vegetative fungal cells, and enveloped viruses. These are some vocabulary terms you guys should become familiar with: sterilization, disinfectant, disinfection. Let's talk through each of them here. Sterilization, note that it is only for inanimate objects. Inanimate means not living, right? So anything non-living can be sterilized. If you imagine, right, the concept here, or the definition of sterilization, is the destruction or removal of all viable microorganisms, all. This means sporicide, right? So eliminating, killing, or removing spores, and then therefore everything else underneath that list. If you think about this, why you can't sterilize your skin or other animate objects, because you are also viable. And so if you can sterilize your skin, you're also going to be killing your own skin cells. So you cannot attain sterilization on animate objects without killing the animate object itself, or at least destroying it quite dramatically. So sterilization is done on inanimate objects. And again, destruction or removal. So there is an ability to physically remove all viable microorganisms from something, and that would be an efficient mode of sterilization without killing them. Disinfection would be one step down, and there are different levels of disinfection: high level, mid-level, low level disinfection, and we'll talk about that at the end of these slides. This is the use of a physical or chemical agent. So physical being heat or radiation, chemical obviously being chemical, to destroy vegetative pathogens. So this is not a physical removal, okay, or that would be mechanical removal of objects. This is destroying them. And this again would be only on inanimate surfaces. When you are dealing with animate surfaces, we are then talking about antisepsis. So for your skin, for example, this would be an antiseptic control, and that's always using a chemical agent applied to the skin to destroy or inhibit vegetative pathogens. Again, destroy means kill, or inhibit means stop them from working. And then the lowest level would be decontamination, which is mechanically removing, like picking something up and moving it away, the mechanical removal of most microbes from surfaces. And this would be either on animate or inanimate objects. So one thing that I like to kind of get you to think about, and actually I'll do this in lecture discussion, but I'll ask you the question here. Let's say that you came to my restaurant and you ordered some delicious meal. Of course, it's going to be delicious. And I asked you, fine, I'm going to go get your meal, but would you like it served on a plate that has been disinfected or decontaminated? And I want you to think about that for a minute. Remember these definitions here: disinfection and decontamination. We'll come back to that in just a minute. Actually, I'm gonna let you just hold on to that concept in our lecture discussion. I'll ask you that question again, see if you can get it. So these are some other ways to think about this, right? And I'm not going to answer these questions here. We'll answer these in our lecture discussion. What process am I using if I clean a skin wound? You should be able to answer that one. What if you spray a counter with a chemical cleaner? What if you're washing dishes in the dishwasher? And this one, be careful, because there may be more than one thing happening inside that dishwasher. How about flaming an inoculating loop, like what we do at the benchtop, or using our incinerators? So those are good ways to think about using those different terms and understanding the levels that are associated with them. We will use these terms again as we get further along when you're deciding which type of chemical cleaner to use in any given circumstance. So this is a chart. I like this one. I think that you guys should, as best as you can, memorize this or be able to apply it. So these are the three different levels of control for microorganisms outside the human body. You can use either physical agents, chemical agents or mechanical removal methods to eliminate microorganisms, either by killing them or inactivating them or physically removing them. And I keep saying physically removal. I'm gonna try to change to mechanically removing them, because that's what your book terms for mechanical. For each of these, there are different kind of subcategories or different types of physical agents here, heat or radiation. For chemical agents, you could be dealing with a gas or a liquid. For mechanical removal, primarily we're just talking about filtration here. And then there are specific examples for each of them. So I think this is from the last version of your textbook. I think maybe they still have this in this current version. They might have modified this some, but I think this is a great way for you guys to really move through this chapter and try to remember each of these categories and when you would use which one. And that's pretty much the biggest takeaway. Like, when would you use each of these types of control methods? Let's start here over on the side with physical agents with heat. So again, there are two different types for microbial contamination. That would be heat or radiation. For heat, you could be using dry heat or moist heat, and dry heat would be variable depending upon the temperature. So incineration would be one level, a dry oven would be another, depending upon the particular thing that you were trying to clean. Moist heat could be steam under pressure or without pressurization, without pressure. And so again, each of these would have a different use depending upon the thing that you're trying to clean. And also note that they have different levels of cleanliness. So sterilization can be attained under some circumstances, otherwise disinfection. So for heat again, moist and dry heat are the two modes essentially, or two basic categories of using heat. Moist heat would be using hot water or steam, either with or without pressure. Dry heat would be the absence of water or steam, right? So this is just air that has been mechanically heated. For moist heat, and actually you can see the mode of action. So the mode of action means how these things work. The mode of action for moist heat and dry heat have similarities. So both of them denature proteins. That means they break the bonds between or within the folding of proteins. But for moist heat, because of the addition of hot water or steam, it also coagulates proteins together, which means clumps them up, kind of like what happens with eggs when you heat them on the stove. So it sticks them together. For dry heat, there's no coagulation because there's no moisture. So the opposite of that would happen, which is dehydration, the removal of water, which is what happens when you heat up air. For both of them, it will kill the organisms through stopping metabolic action, since both of these processes interfere with proteins and macromolecules. For dry heat, you can completely incinerate something using very high temperatures. That's what we use with our incinerators on our benchtop when we're dealing with inoculating loops or inoculating needles, that we actually are trying to completely not just kill the contaminants, but remove them altogether by incinerating them into ash. For moist heat, these are some of the different modes of moist heat. So steam under pressure would be the best mode of cleanliness, or the best way to sterilize an object, as long as it is not heat sensitive or moisture sensitive. Those are the two major things to consider. So this would be using an instrument that we call the autoclave that you see in this picture here. There are lots of variations of this. Some are really, really big. Some are a little bit smaller. Actually, this is kind of on the small end. So an autoclave is a specialized machine where you put in objects that are not heat sensitive or moisture sensitive, and you seal with a tight seal. Usually this requires one of those crank things. We call that a crank, and a really, really tight seal for the instrument door. And this raises the temperature, but it increases the pressure. And if you remember your chemistry, maybe that would be physics class, you can attain a higher temperature with increased pressure. So it brings things to a very, very high temperature without actually destroying the object. And because of that high temperature, this is definitely sterilization. Nothing can survive an autoclave except for prion. Non-pressurized steam would be just putting something over a steam bath. Usually this is not a very good method of obtaining sterilization, but still, because it's high temperature, you might be able to have intermittent sterilization, not usually used very often. Pasteurization is what's done to disinfect, not sterilize, but disinfect liquids for the purpose of bringing the microbial contamination down for consumables, right? So things that people will purchase and consume, eat or drink. So anything that is shelf stable has been, well, let's see, anything that's in the refrigeration section in your grocery store, well, most of those things have been pasteurized. So they've been heated to a particular temperature to eliminate most microorganisms. Now, pasteurization, again, is not sterilization, so endospores could still be in there, but that's usually okay because this is something that you're eating or drinking, not sticking into your tissue. And usually your immune system is sufficient to prevent the growth of spore-forming bacteria in your gut. It also potentially could keep a low level of microorganisms inside that liquid substance. So for example, milk would be a classic type of thing that is pasteurized and put on the shelf, right, in refrigeration. If you ever see it outside of refrigeration, that usually means it's been ultra or ultra pasteurized. That means a very high level, which essentially kills everything. And there's no detectable microorganism life or viability in that substance altogether. So it can essentially stay indefinitely without spoiling because there's no viable life inside there. Now, one of the major problems with high levels of pasteurization for some of these consumables is that not only does it destroy the proteins and enzymes inside the microorganisms, which is good, but it also destroys proteins and enzymes in the liquid substance. So just going back to milk as an example, if you pasteurize or do a high level pasteurization of milk items, you actually destroy most of the, or many of the bioactive molecules in milk, including some key vitamins and enzymes. So it's kind of a trade-off when you pasteurize a consumable, that you certainly are eliminating microbial growth, but it comes at a cost. And then boiling water, this is the lowest level of cleaning a liquid substance, but it certainly is effective at eliminating most viable organisms from that liquid thing, whether it's water itself to make it a potable water, which means able to be consumed, or if it's putting something into that boiling water to clean that item, that fomite. So it still is effective, does not usually, you cannot usually sterilize something, so you can't eliminate endospores, but nonetheless, it's a very effective method of getting rid of most viable organisms. For dry heat, again, the incinerator is, or this incinerator that you see here, this is what we use in the laboratory to completely not just eliminate viable organisms and spores, but also incinerate them or turn them into ash. An oven would be the other mode of dry heat, right? And for ovens, it's not very common that you can sterilize, but you can obtain that if you heat it for a high enough temperature for a long enough period of time. So the question here, when would you use dry heat over moist heat to sterilize? So the answer is simply thinking about the difference in those two environments, dry versus moist, right? One is dry and one is moist. So if you are dealing with a moisture sensitive item, then you may want to do a dry heat over moist heat, right? Something like wood or a material that could actually be damaged with steam, especially steam under pressure, then you would want to use dry heat instead of moist heat. I mean, and you can also go back to this one, think about the incinerator, right? The incinerator is a very, very rapid instrument that we use on a regular basis to eliminate life forms from inanimate objects very quickly in the laboratory, versus moist heat. You would actually need to spend quite a bit of time. It takes about an hour, even for the short cycles, in an autoclave to attain the same level of sterilization as the incinerator. So that would be a consideration as well. Okay, radiation would be the other type of physical agent, and radiation could be either ionizing or non-ionizing radiation. Radiation itself, the concept here is energy that is emitted, so transmitted out from a radiation source. So energy emitted from atomic activities and dispersed at high velocity through matter or space. There, again, as mentioned, there is either ionizing or non-ionizing radiation. And these are used for many different applications, but we're talking about them specifically for use in microbial control. So first, ionizing radiation, the concept again, when a cell is bombarded with waves or particles, the radiation ejects orbital electrons forming ions. So I'll have a picture in just a second to show you what that looks like. This results in damaged molecules altogether. So proteins, DNA, macromolecules, components of the cell wall, et cetera, right? Any of those things that gets hit by ionizing radiation will be damaged and form ions that can be very, very reactive. Examples of ionizing radiation would be gamma rays, X-rays, and high-speed electrons. And this always leads to sterilization. Everything gets completely destroyed. And the key thing here is that ionizing radiation can penetrate anything and everything, almost. I mean, of course, there's heavy metals that it cannot pass through, which is why you wear a lead apron when you're going to the dentist's office to get your teeth x-rayed, or if you go to get your x-ray for your broken arm or whatever it might be, they put a lead blanket essentially over the rest of your body. So there are things that these ionizing rays cannot penetrate, but microorganisms, all of them, and including endospores, are completely susceptible to this type of radiation. So here's a picture of what this looks like. Here's some type of radiating source. And there is energy that is emitted from this radiation source. As mentioned, it can pass through just about any barrier. There's very little exception to this. And when it hits the cell and it penetrates inside the cell, any kind of molecule that radiation interacts with, that molecule will be excited to a degree, to an energy degree that electrons are actually ejected from that molecule. And this could lead to breakages, for example, here, or lethal toxins that are generated that become highly reactive inside that cell. This is as opposed to non-ionizing, which does not penetrate, does not pass through barriers, and also does not create those free electrons. So in this case, during bombardment, the radiation excites atoms by raising them to a higher energy state, but there is no creation of ions here. This leads to formation of abnormal binding within molecules because of that higher excitation of energy, but no generation of ions. The best example of this is UV radiation, which we've talked about already in our chapter on genetics. So you should be thinking already of what we know about UV radiation and the damage that it can cause to microorganisms. Generally, this is considered a disinfectant. As we saw, however, in our UV lab, extended or prolonged UV radiation can be a high level of disinfectant, means kills just about any viable microorganism. But the reason this is considered a disinfectant and not a sterilant, even if it can kill almost every type of viable microorganism, is because it cannot penetrate barriers, which means it cannot penetrate the spore envelope. What do we call the spore? Cortex, thank you, okay. The cortex of the spore, right, is super, super thick, dehydrated, specialized peptidoglycan with extra layers on it. So becomes essentially a barrier for UV penetration. So if there is a barrier in place, and actually we saw this with our UV lab as well, any type of barrier, UV is not a very high energy radiation, so it does not pass through or penetrate through barriers. When there is no barrier involved, it will excite molecules and cause abnormal pyrimidine binding, or thymine-thymine dimers. Okay, so we just talked about the answer to that first question. You can think about that too. And the second question, why might you want to use non-ionizing radiation over ionizing radiation? This is going to become a regular question that you want to ask yourself anytime you're given the option of different types of cleaning methods to use. Sometimes, well, I guess I should say in all cases, it's not just a question of what level of cleanliness you need, whether it's disinfection or sterilization, what type of microorganisms you're trying to eliminate, but it's also a question of the user, right? You are the administrator or the administrator of these different types of cleaning things. And if you, so that should always be considered part of the equation, is how this thing will be administered. And for ionizing versus non-ionizing, that should be evident which one is least toxic to the administrator. And so only under very unique circumstances would ionizing radiation be used to clean some type of surface if non-ionizing radiation is available. Okay, so we're done with physical agents, talking through heat and radiation. Physical means to interfere with microorganisms and cause physical changes, specifically to proteins, enzymes, macromolecules, DNA, etc., different components of that cell, to cause alterations in metabolic activity or reproduction. Over here in green is another category. This is mechanical removal. And really for your textbook, we're just talking about filtration here. That would be HEPA filters for air or membrane filters for liquids. Note that in all of these categories, decontamination is essentially the highest level, and we'll discuss that here. So decontamination is if you recall, physical removal of most microorganisms from something. So again, we're talking about liquid or air. HEPA filters for air. This is what you would find in the ventilation system of every hospital or clinic, ideally, to prevent any aerosolized contamination from one room to another. This, of course, becomes super important with things that have aerosolized contamination, including our dear coronavirus that we're dealing with right now. Even though that's aerosolized particles or droplets, they can be very, very small, even microns in size, and therefore have the potential to pass from one room to the other if there is no filtration system in place. So these HEPA filters are very, very important in healthcare settings to essentially clean the air from one room as it leaves and prevents the microorganisms from contaminating another room. And otherwise, if we're talking about fluids, then we use membrane filters. The concept is the same though. So a filter, whether it's for air or for liquid, is a type of substance. And actually UC Davis has a textiles department where there are specialists that deal with this concept right here with talking about the filtration capacity of different types of textiles, which is really what we're talking about. There are different substances that can create these different membranes. So we use nitrocellulose membranes for different types of molecular biology work, or if we're talking about other applications, there are other types of substances that can be used for these filters. And for manufactured filters or membranes, the pore size can be adjusted depending upon what you're trying to separate. Sometimes, especially for something like the molecular biology work that I do, I get my pore sizes down to very, very tiny because I'm trying to separate out different types of proteins, right? So I have a 300 kilodalton protein filter, for example. And so anything that's larger than that will stay on this side. Anything that's smaller will pass through, and I can collect it and separate those things out. For microorganisms, that's way smaller than is really needed. So most microorganisms are about 1 micron, maybe a half of a micron in size, except one huge category, which would be viruses. So most of the time, filtration filters out everything except for viruses, because if you recall, the size of viruses is really, really tiny. So these are nanometers in size. Now it is possible to develop pores, membranes with pore sizes that are small enough to prevent the passage of viruses, so down to nanometers or smaller than, I guess most of them are larger than 1 nanometer. So about 1 nanometer in size would be the pore size that you would need to effectively sterilize something. But it's very expensive, very costly to develop those membranes. So therefore, most of the time what we're talking about is decontamination, where some viruses still get through, but it is possible to get that high level of filtration. And in fact, there's a whole conversation that the world is having about filtration right now, specifically dealing with coronavirus masks, right? So those different types of masks people are wearing, N95 would be one of the highest levels of filtrations that can be provided, but it's really only effective if you can wear it appropriately to get it to essentially seal off your entire respiratory pathway, so your nose and your mouth. and it's small enough that it can effectively filter out viruses and prevent both the transmission into your mouth, and also, if you are sick, prevent the transmission out of your mouth into the environment. There are different types of masks and filters that people are using, and all of those have varying degrees of effectiveness and have varying capacities for success at preventing transmission. So it's a whole conversation that we could have outside of these lecture slides. All right, so the third category here would be chemical agents. Chemical agents, this is sort of the second half of your chapter textbook, or your textbook chapter, really just talks through a whole bunch of different categories of chemical agents and discusses how they work, so their modes of action, and how that leads to either disinfection or sterilization. And if disinfection, what level, high level, mid-level, or low-level disinfection. Remember that antisepsis is in this category too. So if you're dealing with an animate object. So before we get into the specific categories of the chemical agents, what I'd like to do is take some time to talk through what I feel like is your takeaway message for this. It is important for you to know the different categories of chemical agents, but I feel the most important thing to think about is how do you effectively choose, or I'm sorry, how do you choose the most effective type of chemical agent to fit the needs of the object that you are cleaning, and to think about the protection of you as the administrator of that particular chemical agent. So let's talk through some of these things. I have like a few slides to talk through these general concepts. So these are really broad, abstract concepts to think about. Chemical agents should work really, really fast, so rapid action at the lowest concentration possible. And again, thinking about this as far as you administering this thing, if you're spraying down a surface, you don't want to have to spray down a very high concentration of something, because then inhalation damage could cause toxicity to you as the user. So lowest concentration that is effective very, very quickly would be best. It has to be soluble, which means, well, ideally it would be not in a powder form, right? You can turn this into a liquid form that can be sprayed or poured out to be used to wipe down things or to saturate different substances, fomites, for example. And stable, that means it should stay in solution. It shouldn't precipitate out and cause toxicity at high concentrations. So this term broad spectrum is important for you to understand. We'll talk about this in our next chapter too, and actually discuss it as kind of an inverse concept as we talk about here. When you're dealing with cleaning a surface or skin or a fomite using a chemical agents, we want to make sure we choose something that is as broad as possible. Broad spectrum, meaning covering a wide range of things. This term microbicidal, cidal means killing, and of course microbe means microorganisms or small things. So microbicidal means you're actually killing. That means you're not just halting the metabolic activity. You want to actually kill these things. And again, low toxicity, that means that you want this to be non-toxic to the human, definitely toxic to the microbe, but not toxic to the human. And then broad spectrum, again, you want something to hit as many diverse types of microorganisms as possible, unless you know exactly the organism that you're trying to kill, right? Like you're trying to kill Pseudomonas, and so you can choose a highly fine-tuned type of chemical agent to kill Pseudomonas in particular. But unless you know that that's the only thing that's on that surface or that fomite, then you want to expand the spectrum of activity as much as possible to be the most effective type of disinfectant. Penetration mostly revolves around the substance that you are trying to clean. So imagine for a minute you're dealing with a solid surface that's non-porous. We don't have to worry about penetration. You just have to spray something or pour something on the surface and make sure that that is effective against a broad range of organisms. But let's say as a contrast, you are trying to clean a bed sheet. Bed sheets, if you do the same thing, spray a chemical spray on the surface of the bed sheet, likely it does not penetrate down through the substance to where the microorganisms actually are inside those fibers, and therefore it is not effective. So penetration really does matter. Whereas on one substance, it will be effective. On another substance, it might not be effective simply due to the level of penetration that chemical agent has. Resistance to inactivation becomes important anytime you're talking about animate objects. So cleaning the skin of a human or an animal. Skin has all kinds of different chemicals, as we already know from immunology, and we have things that we produce that inactivate chemicals that are toxic to us. So we have to make sure that, and actually I guess microorganisms also on that same note have different modes to inactivate toxins that are outside of that organism to protect them. So you definitely want to choose something that is resistant to inactivation. Non-corrosive and non-staining, right? So this is kind of just the aesthetics of it. As a user, you definitely want to not stain a surface. You don't want to stain the skin. Non-corrosive would be important. Anytime you're cleaning something, of course, you don't want to corrode that item after or during use of that chemical agent. And again, more aesthetic, sanitizing and deodorizing. And then something that you don't really think about when you're doing academic work, but this becomes really important in the practical use of any of these chemical agents, whereas gamma radiation might be the most effective means of sterilizing the surface of a room. The likelihood of you being able to access that gamma ray and afford the use of that gamma ray, since you need a specialized radiation technician to be able to administer, you're right, so you're kind of getting where I'm going. The academic understanding of these different things might not translate to the practical use of them in a hospital setting or elsewhere, your kitchen. Okay, so some more concepts here. These ones are going to be a little bit more important for you when choosing the most appropriate chemical agent. And I think this is not from your book. So if you're looking at the slide and you're trying to find the correlated parts in your chapter, I think that I took this from a different book, but I really like this. And I want you to be able to use these terms when you're deciding the best chemical agent to use in each circumstance. So instruments, and now we're talking specifically in the hospital setting, instruments have different levels of use, right? So a critical instrument is one that has direct contact with the deep tissue of the body. This is an invasive tissue. The most classic examples would be surgical instruments or needles. You could probably come up with a few more on your own, but those would be the most basic baseline level of critical instruments. Things that penetrate past your skin, past your mucosal barrier, and then they get into your deep tissue. And as we mentioned before, spores love deep tissue. They love the anaerobic environment. They love the moisture and the ample amounts of nutrients, all of the amino acids that are in that area. So spores really do very well in deep tissue environments. So because of that, and of course, because everything else also needs to be eliminated from those surgical instruments to prevent infection, then a high level of cleanliness needs to be done, which is sterilization, the highest level. So all of these items, all critical instruments must be sterilized before and after use. Semi-critical instruments are ones that come in contact with mucosal membranes, but do not penetrate through to deep tissue. So this would be, just think mucosal membranes, going back to our chapter on immunology, we said that really any part of the body that has epithelial lining has a mucosal membrane to it. So that would be starting from your respiratory tract all the way down, your gastrointestinal tract from your mouth all the way through the other end, even your genitourinary tract, all of that stuff has mucous membranes, right? So as long as that tool is being used only to examine those surfaces of your epithelium in any region of the body, you don't really need to worry about spores in that regard, right? You have to obtain a high level of disinfection because you want nothing viable to be on that item, that instrument, if it's going into the body. But as long as it does not penetrate the mucous membranes, we don't need to worry about spores, because spores do not do very well in that area of the body, essentially, because you have normal flora that can eliminate those spores. So you do want to eliminate all viruses and all viable vegetative cells. High level of disinfection for these ones. And then a non-critical instrument would be something that contacts unbroken skin, right? So a stethoscope or even the surfaces would be non-critical down at the bottom of this list. So with the consideration, sorry, I don't know why my animations are out of order, but let's just kind of work through it. Considering the levels of instruments that we just mentioned, critical, semi-critical, and non-critical, now we can go and talk about the potency of chemical agents. So the last slide was talking about the instruments themselves. This slide's talking about the chemical agents themselves. So sterilants, right, chemicals that can sterilize things, treats heat sensitive. And why is that? Because if it was not heat sensitive, then what type of cleanliness or what type of sterilization would you use? Likely an autoclave. That is the best method, unless of course you're out in the middle of the field and you're working with, you know, critically wounded patients in a war zone or something where you have no autoclave available to you. Then you would use a sterilant that's a chemical agent instead of an autoclave. But otherwise, an autoclave is used preferentially to sterilize any type of critical tool, unless then you would use a chemical sterilant. And again, this kills not just microbes, but also endospores, importantly. So if you're dealing with a semi-critical tool, remember we said a high level of disinfection. The semi-critical tools are ones that invade the body, but only to the mucous membranes. So we do need to kill all viruses and vegetative cells and get them off of that substance. So you would use a chemical that has a high level of disinfection. Intermediate level disinfection would be used to treat non-critical instruments like the stethoscope that we mentioned. So this kills all vegetative cells, but not necessarily all viruses. And again, if you're just dealing with unbroken skin, you're going to be okay. You don't want to invade, or you don't want to contaminate the skin with bacteria. Viruses, very few of them actually do anything when they sit on the surface of the skin, so you don't have to worry about that as much. And then if you're just dealing with surfaces themselves, then you would use a low level of disinfectant. This kills only living bacteria, fungi, and only enveloped viruses, not even the naked viruses necessarily, but that would be sufficient for surfaces. Okay. So the rest of these slides are going to talk through each of the categories. I guess we do have time here, so let's just kind of go through this. If you want to, you can stop the video now and just go through your textbook, because this is all pulled directly out of your textbook. And actually, just for a second before I go into these categories, let me just show you from my microbiology class. This is what I would like for you guys to do, is pull up, this is from your chapter 9 workbook, and this is the last page of that workbook here. And this table is on the chemical control agents that your textbook talks about. I think now your textbook has taken all this information and put it into a big table or something. So if you have a previous version, you'll find more information. If you have a current version, you might need to go outside your textbook just a little bit on a couple of these things. But this talks through all these different categories of chemical control agents. It asks what the mode of action, so how do these things work? Asks the level of cleanliness, so is this a sterilant or is this a disinfectant? And if it's a disinfectant, do you know what level, a high, mid, or low level disinfectant? Give an application or an example of each of these things. Describe any limitations that you know about for each of these items. And then the site, so not just how it works, but what part of the bacterial cell or virus does it interfere with? Is it cell wall, cell membrane, nucleotides, proteins, etc. Right? So this is just a place to start off to think about the site of action for each of these things. Usually I provide the opportunity for you guys to use this table on the exam, but please don't go outside and write extra notes on here, right? I just want you guys to be able to use this to categorize all of your information. But if you want me to talk through it, I will talk through it. Here we go. Okay, so let's talk through these categories. Again, hang in there if you want to, you don't have to. Okay, first category would be chlorine. Chlorine. Chlorine, the mode of action, what it does, it oxidizes the sulfur and hydrogen group on the cysteine, which is an amino acid, and therefore it disrupts any kind of protein or enzyme. So it interferes with these disulfide bridges on proteins and enzymes, which cause denaturization of proteins and enzymes. Disulfide bridges are not found on DNA, so this does not interfere with DNA, but they are found on any proteins. My Mister system is dripping onto my keyboard. Okay, the uses of chlorine. We know already that chlorine is added to drinking water. So at low levels, you'll find the stuff that comes out of your sink has chlorine in it. Sewage and wastewater is treated with chlorine to eliminate any type of microorganism that might be in there. Food equipment, pools, wounds, equipment, right? All of these things can be cleaned with chlorine. And of course, bleach is the most common form of this, has chlorine in it at particular concentrations, depending on what concentration your bleach is in. Jeez. Iodine is another type of chemical cleaner, right? Iodine is a really effective antimicrobial because it has rapid penetration. That means it can get through even on skin wounds. It's used very, very commonly. The mode of action, what it does, it interferes with the hydrogen and disulfide protein bonds, same thing as we saw before. This is considered a disinfectant and can be used as an antiseptic. So it's a great wound cleaner because of its ability to kill microorganisms that are there. And it can be used as a disinfectant as well. One of the, of course, limitations for iodine is that it stains, so something to keep in mind. Phenols. Phenols are derived from coal. Their function is different depending upon whether you have a high or low concentration of this. So this can completely disrupt different components of bacterial cells, can inactivate enzymes even at low concentrations. It's considered a disinfectant and can be high level or low level disinfectant depending upon the concentration of this. This is one of the key ingredients in Lysol, which is a derivative of phenol. Triclosan is also phenol is also a major component to triclosan, which is a type of antimicrobial agent that's used in different types of soaps, or at least it was used in different types of soaps until I think the FDA prevented the use of triclosan in soaps because it does not seem to be all that much more effective against microorganisms than non-triclosan based soaps. Chlorhexidine is another one here. This one is considered a surfactant. So surfactants interfere with the permeability of cell membranes, so it targets cell membranes. This, if you can think about it, that means that this would be great against bacteria, but also any enveloped viruses, because they also have that same cell membrane component to the envelope. Lowers the surface tension until selective permeability is lost, and then it can denature proteins as well. Considered a bactericidal at high concentrations, right? An antiseptic for its use. You can find it in some things like this, body washes or wound degermers. And this is a picture of surfactants. Someone just told me what this term surfactant was derived from. It's like surface acting antimicrobial. Surface acting antimicrobial, I think is what it means. But surfactants. So surfactants work, and keep this in mind because we'll talk about this also in our next chapter on antimicrobial drugs. So these guys physically insert themselves in the cell membrane, which eventually causes the loss of that selective permeability and causes the dissolution of that cell membrane to cause the leakiness of this thing, which causes all kinds of different problems. Alcohols. Ethanol, isopropyl alcohol are the commonly used alcohols for cleaning. You can turn this into a sterilant, so a germicidal. It means it really kills just about everything. I guess it does not necessarily kill spores unless you have a very high concentration for a long period of time. So the concept here is that it's an alcohol, so it dissolves membrane lipids, can disrupt surface tension. It's not exactly a surfactant because it doesn't insert itself like we saw for chlorhexidine, but it does still interfere with membrane lipids and causes the disruption of surface tension, and also then can denature proteins. This is non-irritating, inexpensive. It's really commonly used to clean wounds, but of course it has a high rate of evaporation. Hydrogen peroxide is used as a sporicidal at high concentrations. We already know how this thing works because we've talked about this before. Oxygen forms hydroxyl free radicals that become very, very toxic and reactive. So hydrogen peroxide has been used classically as a wound cleaner, but now it's, or I would say has classically been used to kill microorganisms inside the wound, but now it's just used to debride wound, which means to essentially like physically bubble out any kind of particulate matter inside wounds, but not necessarily to kill microorganisms because of the catalase activity inside our cells, right, that can kind of inactivate this molecule. Rapid sterilization of expensive, delicate, and invasive tools, keep this thing in mind. So here's a picture of a cabinet that is a hydrogen peroxide cleaner. This is a sterile processor for endoscopes and other microsurgical instruments that are heat and moisture sensitive, right? So, well, heat sensitive in particular because of the materials that they're made up of. So detergents act very similarly to surfactants, right? So they have a cationic charge for cationic detergents. They bind with negatively charged surfaces, proteins on the membrane or membrane phospholipids can disrupt that cell membrane, just like we talked about before, and cause decrease in selective permeability again. So you'll find this, obviously, you know, many applications of detergent, cleaning agent, sanitizer, soap. Aldehydes like glutaraldehyde there are these large molecules, and they cross-link, means that they bind to multiple proteins and cause them to link together so that they stop functioning like as surface receptors. It's called alkylate, so it physically bonds, changes the place of a hydrogen bond and puts this giant glutaraldehyde molecule in the place of that hydrogen bond, which completely stops enzyme activity. Rapid and broad spectrum, if it's high level or if it's in high concentration, it can be sterilant or high level disinfectant. There are some limitations to this one. Acids and alkalis are used, if you think about this, right, for food preservation. So we use this has been used for thousands of years in human history, that we use acidity to preserve our foods, or we allow organisms to ferment our food items so that they produce that acid byproduct, which prevents the activity for most other organisms. So this kind of goes back to our chapter 6 when we talked about different, or chapter, I don't know what we talked about that in, chapter 6 for acid and alcohols, and then chapter 7 for metabolic activities. So it kind of goes back to everything we talked about. Of course, acids and alkalis, that means positively charged or negatively charged solution. Low pH or high pH solutions are incredibly damaging, not just to microbes, but also to humans. So this needs to be considered if this is the application that you choose. But these alkalis, so high pH enzymes are used in detergents, and also like ammonium hydroxide would be used as a cleaner. And then food preservatives, usually acids, are used for that. Okay, this is how I will be testing you guys on this information. And in fact, for spring 2020, you guys will be having a take-home portion of your exam just focused on this kind of question. So think about all of these microbial control methods that we talked about, all 3 of those different categories. And for the chemical cleaners, all of the different types of cleaning agents that can be used and all the different applications, limitations, and how you would effectively choose which type of cleaner one from the other. And then answer the following questions. So for each of the items on the next slide, think about what level of instrument use that particular item is. Some don't really fit into this question very well, but you'll see that in a minute. Considering what level of instrument use it is, what level of control then would you choose? Sterilization, disinfection, high level, low level disinfection, and then discuss why you would choose that. And then with this information, then talk about which category, so physical, chemical, or mechanical control. And then within that category, what is the best control agent? So an example, if you chose the chemical, if you chose the category of chemical control, then you would go through your whole list of chemical agents and then choose the one that would be the most appropriate depending upon the instrument level and the mode of action and the accessibility, affordability, all those other considerations we had talked about before. So there are wrong answers, right? Or answers that would not be the best choices. And when you're answering these questions, don't always just go directly towards chemical control agents just because you know about those. Think about the best type of control, physical, chemical, or mechanical first. And then once you choose that, then you can go in and figure out what the best exact control agent that you could use for each of these categories. And then add one more question on here, discuss the mode of action. So how does that example that you provide work, right? What's the mode of action? What does it do? Not just in general, oh, it kills all microorganisms, specifically how? Does it denature proteins? Does it bind to, does it replace hydrogen bonds and add a glutaraldehyde molecule to interfere with protein activity, right? So get specific here on the mode of action for each of these things. And so this is the list I have for you just as some examples. This is definitely not an exhaustive list, but I'd like for you guys to just start practicing with these ones during our lecture discussion time. I'll give you a little bit of time to work with your partners on this one and just prepare yourself, because this is the type of question that I'll be asking you on your take-home exam. Okay, and that's it. That whole chapter is done.